the third lab overview in terms of things you need to know to better understand your data. Now, before we get into our lesson, we're going to talk about some ideas that we've already learned this year that might be really useful to help you think about why this lab worked and what data is showing us. The first is that when we talk about enzymes, we need to remember that proteins, which include enzymes, are long strands of amino acids, hydrophobic, hydrophilic, positive, negative amino acids, where R groups interact to make a very specific shape, and ideally then a very specific function. Now that shape and function is really important for enzymes, in which the enzymes have this place, this location called the active site, that allows them to bind with a substrate and break it apart. Specifically, there's always a catalytic site, which is a chemical location, and the binding site, which is the physical location that breaks apart our molecules. Another really important idea is that cells will make proteins in response to a signal cascade, or what we learned over our second unit as the 3 steps of cell signaling. There's always a reception, then transduction, and then finally the response, which is often the expression of a gene to make a protein. And then the last idea is that atoms are balanced with 8 electrons within their valence electrons. So atoms need 8 electrons, and that makes them balanced, and hydrogen needs 2. And that makes them stable. So let's get into the first idea. Now, when we talk about cellular respiration, it's always going to be when organisms react glucose with oxygen in an enzyme to make some CO2, which we measured in our lab, water vapor, which is why when you breathe outside and it's cold outside, you see mist, and then finally ATP, which allows cells to do work. Remember that phosphate molecule makes proteins turn on or off. This is an exergonic reaction, which means we're taking a lot of energy stored in sugar to make all of these things happen. Now, monosaccharides are simple sugars in that they contain one hexagonal shape. So this 6-sided structure represents the 6 glucose molecules, or 6 carbon molecules, bound together in the sugar. Disaccharides are molecules that have 2 hexagonal structures linked together, and polysaccharides have many more monosaccharides. So in order for us to react disaccharides or polysaccharides to make ATP, we have to break them down into smaller pieces or smaller chunks. So let's talk about the different sugars that we had. We all used starch as one of our sugar sources, and starch is a polysaccharide, which means it's a long chain of glucose molecules, monosaccharides, all bound together. In yeast cells, they use an enzyme with an active site called amylase that breaks apart this starch molecule into something smaller, because right now this starch molecule is too big to react. It's like putting a giant log in your wood stove and expecting it to catch on fire without any kindling. Amylase is going to break down that starch into a smaller molecule known as a disaccharide, which is maltose. Notice we still have 2 glucose molecules bound together. Well, this molecule is still way too big, so we're going to have to react it with another enzyme to break it down into a monosaccharide. So we use the enzyme maltase that binds to maltose, and it breaks it down into our monosaccharide glucose, which is also known as dextrose. This glucose, in the presence of oxygen in an aerobic environment, will react with zymase to make our CO2, our water vapor, and our ATP. Now, there are other disaccharides, other 2 sugar molecules that we reacted with in our environment. Another one that we've been using for all of our labs is sucrose, or table sugar. That is made up of a glucose and another monosaccharide known as fructose. When we have sucrose molecules, we use an enzyme invertase to break it apart into glucose and fructose, both of which can be reacted with by zymase to make CO2 and ATP. So if we break apart maltose or sucrose down into their monosaccharide components, this enzyme zymase will make our CO2 and our ATP. If dextrose or levulose is by themselves already to begin with, it's a 1-step process to make CO2. And then finally, we also added lactose. Lactose is a disaccharide, which is glucose and another monosaccharide known as galactose bound together. You need the enzyme lactase to break apart this molecule into our monosaccharides. However, lactose really isn't made by yeast. It's made in very small amounts, which means it's a not very digestible sugar. It doesn't make a lot of glucose and galactose because this enzyme is missing. So again, in a nutshell, the bigger your carbohydrate is, the more steps you have to take to get it into a monosaccharide form so that we can make CO2 and ATP. If you're already in a monosaccharide form, you're going to be able to be reacted instantly to make CO2. And the reason why we use all of these different enzymes is when we look at the structure of disaccharides, whether it's sucrose or lactose or maltose, they all have slightly different chemical structures, especially within the orientation of their bond and the molecules that surround them, which means we need different active sites with different amino acids to interact with those positive and negative regions of those sugars to break it apart. I need a house key to break open my house, I need a car key to start my car, and I need a bike key to unlock my bike lock. My bike lock will not be able to start my car. Now, this is all contextualized, these enzymes, within the fact that yeast are living organisms. And enzymes are proteins that need to be made by cells for them to undergo these metabolic processes. They need to make these proteins first to be able to react each of these sugars. Now, within our cells, we have our nucleus that contains our chromosomes, which are made up of DNA. This DNA is the instructions, the information that cells use to make those proteins. And proteins have a really specific sequence, right? Ethan versus ethna are two different words that have two different meanings. One of them is correct and the other is incorrect. DNA tells the order in which these proteins to make their amino acids. So genes, which are part of our chromosomes, contain the information for proteins to be made properly. How do they do that? Well, DNA is coded by these things known as nucleotides. And nucleotides come in 4 different flavors. I need to move this slightly right now so we can see it. They come in, a code that's either big or small. So 2 of our DNA nucleotides have double structures. 2 of them are single. And also they either have 2 hydrogen bonds, positive negative interactions, or 3. So when cells read parts of their DNA to make a protein, they get a code that might be big 3, small 3, small 3, big 3, based on whether or not there's 2 hydrogen bonds and whether the A or the T is big or small. Each gene that makes each enzyme has a very specific code. Sort of like when you open a recipe book, each page has a very specific recipe that uses a very specific set of ingredients with a very specific set of procedures. What this means is if a cell has a gene for amylase, it can't make maltase. And alternatively, if a cell has a maltase gene, it's not going to be able to make amylase with that gene. One gene can be only used to make one specific protein. So if a cell doesn't contain the gene to make a specific protein, it's not going to be able to have the instructions to make that. It's only going to be able to make the enzymes that it has the genetic code for. So because lactose doesn't have a gene for, or because yeast don't have a gene for lactase, they're not going to be able to make that protein. You can only make the proteins that you have the genetic code for. And again, we read DNA as either big or small, 2 or 3 hydrogen bonds. That's the code that we read to make our proteins. So let's talk about the last key idea, which is acids and bases in pH. So when you look at your data results, pH, whether it's 2 or 6 or 10, It's just a measurement of acidity or bases in a solution. A solution that's an acidic solution means that it has a high number of acids, and acids are hydrogen ions that lack an electron. What that means is that these hydrogen ions want to go out and react with electrons to make themselves neutral, because currently this is positively charged. An acid is just a positively charged hydrogen atom that's missing its electron, and it's going to steal electrons from other molecules in order to balance itself out. Alternatively, a base is a molecule that contains a disproportionate number of hydroxide ions, which is water missing one of the hydrogen ions, but it has an extra electron. That electron makes it have a negative charge, which means that this hydroxide ion is going to try and bind with hydrogen ions to balance itself out. So when you react an acid with a base, you have a chemical reaction where the two things equalize. Think baking soda and vinegar. Poof, you get a volcano. These two molecules, though, separately will be reactive with other things to try and achieve a balance. So generally, positively charged hydrogen ions will go for negatively charged molecules to get those electrons, and hydroxide ions will go for positively charged things to get that extra, or get rid of that extra electron. Now, when we talk about what these pHs mean, we can look at the scale to get a better idea. A pH of 7 is considered neutral, and that's because there is an even amount of hydrogen or positively charged molecules and hydroxide ions, which are negatively charged. If we go below 7, we have an increasing number of H plus or positively charged molecules. In each step up, so from 7 to 6, or 6 to 5, or 5 to 4, is a tenfold increase in the number of acids. So if you compare the number of acids in your saliva compared to distilled water, there's 10 times as much. But if we compare black coffee to distilled water, there's 100 times more acids in black coffee than there are in distilled water, and 10 times more acids than in saliva. And the same thing is true for bases. If you go below a pH of 7, you have an increasing amount of hydroxide ions or negatively charged molecules. And every change down makes it more basic by a tenfold value. So there's a tenth less acids or 10 times as much bases in seawater as compared to distilled water. If you look at soapy water, which has a base of 12 from all of that detergent, it is 100,000 times more basic than distilled water, or 1/100,000th times less acidic. So each of these is always a drop in 10. And remember, acids are positively charged and will react with negatively charged molecules, and positively charged molecules will react with hydroxide ions because they're negatively charged. This might be important because, as we learned at the beginning of our second lab, enzymes have positively and negatively charged regions in their active sites that allow them to bind to substrates. So it might be that acids and bases do things to enzymes and their active sites that might change their structure and function. I hope this video has been helpful. We'll of course talk about these ideas on Monday and Tuesday of next week. Feel free to come in with questions, but again, with your notes, prioritize pictures and explaining those pictures.